how the image is formed in human eyes. All of our eyes, we are actually visualizing everything around us, and in doing that our eyes does the job of formation of an image. Without the aid of microscope also we are able to view, and how basically our eyes works, let's see that. When we are trying to look at something that's very far from your sight, let's say this is the eye lens that you have. I'm representing it as EL, and in this EL lens, if I actually am observing a bird which is flying in the sky, which is very far from me, right? If I'm going to observe that bird, for me, that object is actually subtending an angle on my eyes. That angle which is subtended by the object onto your lens of the eye is basically referred to as alpha, which is shown here. Now, when we are observing something that's very far away on the lens of from the lens of the eye, it's going to subtend a very small angle, and hence the image that is formed on our retina, the resultant image that will be formed because of the subtended angle on our retina, will be very tiny. That's why when you see an object which is very far away from you, you see it very tiny. However, as you bring it closer, let's see what happens. While when we are observing something that is very close to your eyes, the angle subtended by the object becomes larger, like this much. So the angle which is subtended by the object onto the eye lens of your eyes is going to determine what is going to be the magnification, or what is going to be the size of the image that is going to be formed onto your retina. Now, when you are actually taking it towards you, the object is approaching towards you, you can see it getting bigger and larger, as a result of which the image formed on the retina is also larger. So the crux is that the size of the image formed by our eyes will be determined by the angle that will be subtended by the object on our eyes, which itself depends upon the distance of the object from our eyes. I hope we are clear with these two scenarios as to how our human eye and the image of the size of the image which is formed in our human eye with respect to the angle subtended, which in turn itself depends upon the distance of your object from your eyes. Now, we have to remember one thing here, is that the focal length of your eye. What is the focal length? The focal length is basically the distance between the focus point, that is the back focal plane or the front focal plane of your lens, eye lens, and the optical center of the eye lens. Now, we have to remember one thing here, that the focal length of our eyes, for that matter of fact, or any vertebrate basically, is very much variable and adjustable, all thanks to our ciliary muscles. We have a set of muscles which can actually regulate and adjust the size of your basically the eyes. It's constantly trying to basically change the focus at which your eyes can look, the eye lens can look through, or whatever you are trying to visualize, you're basically going to adapt it very soon. Your eyes are basically adapted to it very soon. Let's say you are running and you're trying to chase something. In doing that process, you never lose the focus of that object if you're going to continuously looking at that object. Why is that happening? Because whenever you are actually running, you're taking your lens and approaching towards that object closer together. So ideally your focus should change, right? However, it does not change because there are ciliary muscles which are in action right now in your eyes when you are running and approaching something, which keeps the focus always managed. Hence, our eyes are having a very nice ability to adjust and accommodate to anything we are trying to see, whether we are approaching this object or whether that object is approaching us, or whether we both are approaching towards one direction. Now, when I say that, obviously it's not unlimited, that our eyes do not have an unlimited capability of focusing it and doing it. We know we can actually read and write only if we are holding our pen and paper at a particular distance. If you're going to take it too close towards your eyes, the vision of your eyes gets blurry. Now, why would that happen, right? Because there's something called as least distance of distinct vision. What do we mean by that? If you further bring the object closer towards your eyes, you will find it very difficult to visualize. the least distance at which our vision is most distinct is referred to as least distance of distinct vision. And for our human eyes, this least distance of distinct vision is somewhere between 25 centimeter. This is the value where the eyes does not have to do any kind of extra adjustment. Your ciliary muscles are not at work, and at 25 centimeter, any object is crisp and clear to you, like it is not when it is away or below the 25 centimeter value. Now let's draw an analogy here. When we are playing cricket game and a ball is coming to you, approaching you with a very high speed, your eyes tries to adjust your muscles. Your muscle is basically trying to adjust your eyes so that all the while your ball is approaching towards you, it will look very sharp to you and you can focus on that ball and hit a shot. Now, not just that, imagine a vertebrate like a cheetah which runs at such a high speed to catch its prey in jungle. Now, before it starts to hunt on its prey, it will actually just lock its target and it's going to lock its vision on the prey which it's going to catch. Now, such that when it runs towards it with very high speed, its eyes manages to keep the prey focused every other second, and even though both the prey and predator are approaching in some direction, because the prey is trying to actually escape the predator and the predator is trying to actually catch the prey, both are moving at a very high speed, and at this moving speed also, throughout it is of utmost importance for the cheetah to catch the prey, and it does because its ciliary muscles of the eyes, and there are certain physiological adaptation in the cheetah which makes it highly capable of its eyes highly capable to see its prey at complete focus even when it's approaching the prey at a very high speed. Now, I actually encourage you all to go and see what is that physiological adaptation in cheetah that's going to lead to such kind of adaptation in its eyes and allowing it to keep the prey at a complete focus even when they're approaching at a very high speed. Okay, so I hope we are clear how image is formed in our human eye and how our human eyes are extremely adaptable to whatever we see in terms of how we focus it, whether it's be close to us or whether it be it away from us. Now, we are aware that the angle alpha, which is basically the angle subtended by the object on our eyes, is important for the image formation. Now, what happens usually is that you keep a lens in front of your eyes and you are trying to see something. Now, what happens usually is you're keeping a lens in front of your eyes. In case of upper illustration, you can see your eyes are there and there is an angle subtended by the object, which is basically determining the size of the image formed by the lens onto your retina. Now, in the lower illustration, if you can see, this is basically your eye lens, which we are showing here. Beyond the eye lens, there is retina, which we are showing here. And on the retina, this image is going to be formed. Now, there are two focal points here. You have a front focal point and a back focal point. However, in this lower illustration, we have introduced another convex lens in front of the eye lens, which is referred to as this. This is basically another convex lens which I am keeping in front of my eye lens. And I will use this lens to basically visualize. So how is the image formed? Now, for this convex lens, we are keeping the object inside the focal plane, as a result of which a virtual image of this object will be formed by this lens. Now, what happened? If this virtual image will act as an object for our eye lens, now you might ask why the object for the convex lens cannot directly act as an object for the eye lens, because when we are keeping this object here, it can also possible that this lens try to image that object. However, it is not possible because the convex lens which collect the light from it, the light has to pass through the convex lens and reach the other lens so that it can be imaged. However, that does not happen. The light cannot reach our eye lens. Because it is always already captured by the other convex lens that you are placing before it. Okay, so if the virtual image act as an object for your eye lens, what will happen? Since this is a virtual image, it's very far away from your eye lens, and the angle subtended by this huge virtual image come object for your eye lens will be large, leading to a overall magnification of your object when you see your through your eye lens and visualize it. Now in this scenario, both the lens are working together to actually get you a magnified image as compared to what you will see otherwise. Now let's again see what is happening here. When I kept my object inside the back focal plane of my convex lens, this is the distance at which I kept my object. Since it is the inside, and when I trace the light path, I actually see that I have to extrapolate the light rays towards the same direction, and I see an upright and a virtual image being formed. This is the virtual image being formed here. Now this virtual image acts as an object for your eye lens. Since it is very far away from your eyesight, this large virtual image or object of your eye lens is going to be formed an image on your retina, which is to be eventually formed here. Now, I hope how the ray diagram worked and how we actually produced the image by keeping another convex lens with our eye lens is clear to you. But now in this scenario, both the lenses are working together to actually get you a magnified image as compared to what you will see otherwise, and that's what is happening in this case. You can see when you are only using this eye lens to visualize, the image was much smaller as compared to what you can actually visualize when you are actually using another convex lens to see it. Now, this is what actually happens when you move the magnifying lens back and forth in front of your eye lens. So how do we actually calculate the magnification for this? So basically this is the scenario of what happens when you keep another convex lens as a magnifying lens in front of your eye lens. Now, larger the angle subtended between the virtual image and the eye, larger will be the magnified image formed on the retina. Now, we know we have to calculate the magnification which is going to be associated with this kind of an image formation. Now we know that the least distance of distinct vision, which is 25 centimeter and represented by D here, has to be taken into account when we are actually calculating the magnification, because it is our eyes we are dealing with here, which has a certain distance of distinct vision. Now, therefore, the distance at which your image will be formed, or V, will be equal to minus D, and hence when you wish to calculate the magnification, the formula gets modified something like this. Now in this case we can in this case we can see that what is the magnification when the object is inside the focal plane. But what happens when we keep the object at the focal point, for which magnification can be calculated by the distance by focal length, and this is the formula when you can use actually to calculate your magnification again when you keep your object at the focal point. I hope we are clear with this also as to how a magnification by a single convex lens in the form of a magnifying glass works for us, and how it associates basically along with our eye lens and form a magnified image which you would otherwise not be able to see, because the image formed by the only our eye lens would be much smaller than what we are able to recognize here. Okay, so let's now move to a microscope system. Now when I say a microscope is going to do a same job for you. It's going to magnify an image. Now, a microscope can be as simple as something like this, and it's simply called as a simple microscope, and we are going to see how it magnifies your object. It is basically in developmental biology labs. Previously, these microscopes were routinely used in such labs for the purpose of dissection of your model organism, which you are going to study in your research to answer your research question. Now, Drosophila like Orchic embryo or C. elegans like model organisms are ideal to be dissected under these simple microscopes that you would see in like a decade before. However, now this has become very less common in these labs, because these labs also have started to use a compound microscope which has many lenses in it, and it's a complicated system like that because it's more commercialized right now. Okay, what a simple microscope, which basically is something which you are seeing on the screen. It has a mirror here which play an important role to reflect the light from your light source and Then you have a biconvex lens to focus and magnify your object. Biconvex lens will focus and magnify your object, while your mirror is going to actually take the light from the light source and actually reflect it towards you so that your sample is illuminated. Now, we have to actually focus and magnify the object. We keep the sample or the object beneath the lens and observe from the top to perform the dissection. So from here you are going to actually see, here your eye lens will be there, this is the convex lens, and this is where your sample will be. And if you want to do dissection, this is basically the stage where you can keep the sample and do the dissection. Now there are certain other components associated with this microscope, and it's not a complex assembly, believe me, because they're much complex microscope we are going to see now and how they work also. In this stage where you're keeping the sample and observe, it has an adjustment knob or an adjustment screw associated with it, and all these parts are basically holded by a metal stand to actually keep every part in a place. And eventually you can use this simple setup to actually dissect your model organisms like Drosophila, and the magnification that you can achieve with such a simple microscope can be calculated as something like this. Okay, so after looking at how assembly of a convex lens, a simple biconvex lens, and a mirror can lead to formation of such a simple microscope, we will see how a compound microscope is going to look like. Okay, now moving to another compound microscope. Now these kind of simple microscopes are rare to see in today's laboratory. So instead, what you actually see is a modern day compound microscope which works and generate an image by means of the collection of lenses. When I say collection of lenses, it is not just a single lens or a mirror, single mirror. It is basically multiple lenses which are working in association with each other to actually produce an image for you, which is basically a job of a microscope. Now, for a reason that previously seen microscope contained just one biconvex lens, that's why we were calling it as a simple microscope. And now when we have a microscope where there are several lenses working together to produce a single image, a single resultant image, hence in its working, since it is complex, is it referred to as compound microscope. Now when we say there are more than one lenses or a collection of lenses or a bunch of lenses, let's see what are the type of lenses we have in here. Now in this lens, in this setup, the first type of lens that you're going to see is an objective lens, which is represented by O. Now why it is referred to as objective lens? The immediate question is going to come like that. It is basically the lens which try to focus the specimen or the object, or it is the lens that sees the object specimen first in this setup. That's why it is referred to as objective lens. And the lens just after the objective, which your eyes will be actually using to visualize the image, is referred to as an eyepiece. It is shown here. Now E is represented as eyepiece, O is your objective, and these are the two primary lenses in your compound microscope which will be actually playing an important role to form an image. Now the lens after the objective is referred to as eyepiece, but there is another lens after the eyepiece which is your eye lens, because you are going to eventually visualize your image through an eye or through a camera. Now in case of when you are visualizing it through eye, this eyepiece comes into the picture, and there is another lens, that is the eye lens, which is there, and you basically the image will be formed on your retina, which is represented by R, and EL means the eye lens. Now in the optical drawing you can see another lens which is basically your eye lens, and behind the eye lens is the retina, which is showing here, and the image will be projected there. You have to take a note here that don't confuse between the eyepiece, which is shown as E, and the eye lens, which is shown as EL. Now basically the fact that we know here what are the different type of lenses, we are now aware of it. So now let's see how these lenses are arranged and why they are why these lenses are arranged like that. But before that, there are some other markings which we would need to introduce to. There's FO and there is F-O, which is basically the back focal plane of the objective, while F-O is the front focal plane of the objective. Similarly, an FE and FE dash is basically the FE is the back focal plane of the objective, while FE dash is the front focal plane of the eyepiece. Now, let's see how a magnified image is formed by this compound microscope. Now, can you all see this tiny object placed outside the back focal plane of the objective here? Let me actually mark this for you. This is basically the object placed here. It's extremely tiny in its size. I hope you are all able to see this, and if you are not, just remember wherever I am marking the arrow to actually show you the place where the object is placed, that's where the object is. Now, it is placed beyond the back focal plane, but it's not too far away from the back focal plane of the lens, the objective lens. Now, because it is outside the back focal plane of your objective, it is going to produce a real and inverted image after the front focal plane. Now, if you want to check how that happens, just go back and see how we traced the three important optical rays and how we actually traced where the image will be formed. So we know from that is going to produce a real and the inverted image after the front focal plane of your objective. Now, in a compound microscope, the relative arrangement between the objective with respect to the eyepiece is made such that the real and the inverted image that is being formed by the objective lens lies within or falls within the back focal plane of your eye lens. What I'm trying to say here is your objective lens is trying to image an object. It produces an image here. Now, I'm saying that in a compound microscope, you arrange the eyepiece with respect to your objective such that this image which is going to be formed by your objective always falls inside the back focal plane of your lens. I hope we are clear with that, because that's where we are going to again lead our image to. Okay, so when your image, the first image is formed inside the back focal plane of your eyepiece, this inverted image from the objective is going to be perceived as an object which is placed inside the back focal plane. As a result of which, what will happen now? It will again generate, now this eyepiece will take this image as its own object and it will generate an image in response to this inverted and real image, and it will produce a virtual and magnified and upright image which can actually now be visualized with your eye lens. So this image which was formed is taken by this eyepiece, and this eyepiece is going to magnify this image further, and it's going to magnify this to produce an image somewhere here which will be basically an virtual image, and it's an upright image which we formed towards the same side of your eyepiece where your object is placed. Now, since this image is such a huge image, which we basically know that virtual images are very highly magnified image, this highly magnified virtual image is going to act as a object which will be taken by the eye lens, that is this eye lens. It's going to perceive this, it's going to see this image, and it is going to project it on your retina. Now, a quick recap. Let me first clear the ink and we will once again see what we saw now. Now, what happened is that you kept a tiny object behind the back focal plane of your objective, which formed an inverted and real image which fell inside the back focal plane of the eyepiece. Now, since it is inside the back focal plane of the eyepiece, it will produce another image in response to it, which is going to be an upright, virtual and magnified image. Now, since it is a highly magnified image, it can be seen through your eye lens, which is projecting again it onto your retina. Now, if you see very carefully here, you would have noticed that the virtual image which is formed is magnified and projected on your retina, finally as an upright image. For an upright object, you get eventually an upright image on your retina. But that is a problem. I will give you 1 minute, you just pause your video and think why would that be problematic. Okay, why would it be problematic is because if your retina has an image upright of your upright object, your brain is going to eventually confuse you and invert that image while you are actually perceiving it. This is the job of the brain to do. It does it like that only. But that's an issue for us, right? Whenever you are going to, if this kind of a microscopy system is going to be used, all the time you are going to end up with an upright image on your retina, which will eventually be perceived as an inverted image by your brain. Hence, in order to get rid of this issue, you need to introduce another lens in between that makes the image on your retina inverted, and then your brain will perceive that inverted image formed on your retina as an upright image, which is basically the correct orientation of your objective as well. I hope this was interesting to you. In this illustration, the purpose was to show the light path or the optical trail which is followed between the collection of different lenses in your compound microscope. Now, if you have correctly understood of how image formation takes place in compound microscopy, I would want to actually introduce another important concept here. But before that, if you have any doubt in this, go back and rewind this video and see and get this concept thoroughly as to how an image is formed eventually on your retina by using a compound microscopy like setup of these lenses. Okay, now when we said that the distance between the objective and the eyepiece is maintained such that a real inverted image always fall within the back focal plane of eyepiece, and this fixed distance arrangement that is maintained between the objective and your eyepiece is referred to as an optical tube. Now this fixed distance arrangement is known as optical tube between the objective and the eyepiece. Okay, I hope with this introduction about how the image is formed in a compound microscope, we are crystal clear with the concept of how the image is being formed in a compound microscope, because this is very important to understand the next concept we'll be discussing, which is known as infinity corrected optics. Now, what do I mean by infinity corrected optics? Now, I am aware that this illustration might seem very busy to you at the first sight, but let me take you through it one by one and at a time. When we compare this illustration from the one which just above it, you can see that there is an objective lens here, which is exactly the objective lens here. You have an eyepiece present here, and you have an eyepiece present here. You have an eye lens present here, which is basically the lens in your eye, and also present here. And again, you have a retina above here, which is also present here. The only difference that is evident to you basically in this illustration is the presence of another small lens which is marked as T here, which is known as tube lens. Now this tube lens is the hero of our picture today. It's basically going to lead to infinity correction optics, which is basically a necessity in your modern day compound microscopes. Okay, after you have noticed what are the differences between the illustration above and below, the only additional element you can see here is the tube lens. In the modern day compound microscopes, the microscope manufacturers have placed this tube lens in a particular position. Now the question arises as to why it is kept like this, right? In this illustration, if you also noticed one thing, that the object that you are keeping is basically kept here. The object that you are keeping is exactly at the focal plane, the back focal plane of your objective, which was not the case in case of your upper illustration where you are actually seeing the compound microscope image formation. However, this necessity of keeping an object at the back focal plane arises because of introduction of your tube lens. Now, the immediate question that one would ask is, why would you keep something like this in your optical setup to confuse and to complicate things? Now, when you are actually keeping your objective at the objective back focal plane, what will happen? If you keep the object at the back focal plane, we will get a parallel ray of light which will meet at infinity, Now if these rays meet at infinity, you will never get the image. But now comes the interesting part, that if you keep the tube length such that this tube length will converge this parallel beam of light coming from this object. So these are the parallel beam of light which are coming from the objective lens. And if you keep the tube lens here, this tube lens is going to basically converge your rays at one point where this image will be formed. Now again, this image is basically formed and the distance between the eyepiece and the objective is maintained such that the image that is formed is inside the back focal plane of your eyepiece, not the eyepiece. Now the rest of the process remains the same. This real and inverted image will be magnified to form an upright image which will be visualized by the eye lens and projected on your retina. Now again, the question is not very clear to us right now as to why do we need to do this complication to our system. Why can't we use the system that we already saw? Why do we have to put this tube lens and complicate our system, and what could be the purpose of putting this in terms of practical application? Now, this tube lens is basically creating a space where a parallel beam of rays are traveling, which we refer to as infinity space. So the question now is again, what would be the application of creating this infinity space in our optical tube where your fixed distance arrangement of objective and your eyepiece exist? So the answer to this question is basically, if you would have seen a modern day microscope, a compound microscopy setup, basically you would see that it has a lot of additional elements into it that we need to be incorporated for specific type of imaging your sample requires. Now for example, of certain optical element that you need to introduce, and you should hamper your optical trail as well include your polarizers and specific filters. Now the introducing all of these different optical elements which are actually necessity for you to do different kind of microscopy in today's world is going to complicate your optical trail and entire optical arrangement very much. How to get rid of this problem? Because you cannot compromise of not using these polarizers and specific filters. You have to use them, but you have to use them such that your optical trail and optical arrangement of the optical elements is basically unhampered. Now how do you do that? In the infinity space where the ray of light are parallel, if you keep any of your optical element in this arrangement of the parallel beam of light, it will not affect your optical train of the microscope, as a result of which your image will still be formed as it was forming as depicted in the above illustration. It will still be formed like that. However, in this case where you will be putting additional filters, you can actually modulate how you see the image and what you see in order to do the specific kind of light microscopy for such microscopy techniques include phase contrast microscopy, differential interference contrast microscopy, or fluorescence microscopy, in cases where you need to fuse some phase shifters or polarizers or specific color filters which will be actually responsible for the image that you're going to do for the imaging that you need to do for a particular kind of microscopy. And introduction to all of these different kind of optical elements has to be done in the kind of the infinity space which is created between the optical tube, in between the objective as well as the eyepiece. Since a microscope is not used just to magnify the object, it is also equally important to visualize the object with utmost clarity. And now when we are going to see different kind of microscopy and contrast enhancement methods in light microscopy, we will see what all different kind of optical elements are actually required to introduce for doing this specialized contrast enhancement in your light microscopy setups. And at all those points, those elements will actually be very important to be introduced in your infinity space. And introducing these optical elements in the infinity space where the light is traveling in the optical trail remains affected by introducing these optical elements. As many optical elements you want, you can introduce in that infinity space. It's not going to affect your overall optical trail of the compound microscopy. And this is basically the example of how a modern day microscopy method and advancements in modern day microscopy methods has lead to development of a complex compound microscopy setup which can basically visualize all the samples that you have with good magnification and a nice resolution. Okay, now that we are clear with this, let us actually see how a modern day microscopy uses this infinity corrected optics. In this illustration again, we are seeing it that at 25 centimeter, which is basically the least distance of distinct vision. In this case, you have an objective here, a tube lens here, an eyepiece here, an eye lens here, and the image is going to be formed on your retina here. Then object you are placing somewhere here, right? You have placed the object here. It is basically your objective lens, basically is going to image. And since it is placed exactly at the focal length of back focal plane of your objective, the rays which will be emerging from the object will be parallel rays. Now these parallel rays will be converged by the tube lens onto the inside the back focal plane of your eyepiece, which will be leading to form a real and an inverted image here. This real and inverted image will then be visualized or magnified by the eyepiece to form a image which is formed here, which is an upright and a virtual image. This upright and virtual image is so magnified that it can be easily visualized by your eye lens. Now your eye lens is going to focus this on your retina, where the image or the final image will be formed and it will be perceived by your brain. Okay, I think with this quick recap, we all now know created in the modern day compound microscopy setups and what is the importance of it. Now, we also know that the total magnification of your compound microscopy setup will be determined by the magnification of all the lenses that you are using. Now you have an objective lens which has the greatest magnification in this case, and you also have a magnification associated with your eyepiece. Now, in total magnification is basically the product of both of these magnification, because when your light is being visualized by your eyes, it is actually being visualized through your eyepiece, which is the lens after the objective, to visualize an image on your retina. But if you're actually not using your eyes to perceive the image, and you are using a camera, in that case you don't have to consider the magnification which is being included because of the eyepiece. In that case, only the camera, basically the objective magnification, is only taken into account because the eyepiece is not in use. And when you are visualizing to camera, that is the case. Now we know that the objective as well as the eyepiece comes in different magnifications. You can see that an objective can have a magnification of 2.5x, 4x, 10x, 25x, 40x, 60x and 100x. Now all these different kind of magnifications are available to you. However, not everything is of use to you. Similarly, eyepiece also is available in different type of magnifications. It has 10x, 12.5x, 15x, 20x and 25x. Now there's something which we call as empty magnification. There's a phenomena of empty magnification, concept of empty magnification in microscopy. Now what is this empty magnification? Empty magnification is basically in which the images are enlarged, or basically when you use higher combination of all these different objectives and eyepiece, you are going to try to aim as to get how much magnification you can get achieve basically with using high magnification objective, right? But that's not true. If you're going to use that, it's not going to be a meaningful magnification. What I mean by that is basically it is useless. It's not going to resolve your image any further. It's going to just simply enlarge your image without resolution, and that's of no use in case of microscopy. Believe me, it does not help you at all. Now, in this table is basically shown to help you define which is basically a good combination of the microscope objective versus the eyepiece. So you can see here we have good representations, good combination represented by the X or a cross kind of a symbol, and you can see what all basically are referred to as good combination rather than a empty magnification. Basically a good combination are those which will actually give you meaningful magnification and better details as compared to what you were seeing in the previous combination, which is just a lower combination below it. Now it will also show you what combinations are basically giving you empty magnification and are of no use to you, no meaning to you in case of microscopy. Therefore, using those magnification combinations is going to waste your time, it's going to waste a resource, and it's going to be of no use to you. Okay, now after looking at all the things about how a microscope is going to form an image with a collection of lenses, let's see how a modern day compound microscope will look like. This is the image of an inverted modern day compound microscope, in which you see different optical elements being actually put into labor, and we'll go through all of them one by one. So the first element in the microscope itself is basically your light source, which is basically going to give you a light in all the direction. Now after that, you have somewhere here, you have a collector lens, which is not marked here. I'm going to write it like this, a collector lens, which is basically going to collect the light from your light source, and it's going to pass it further on. Now, in order to control what amount of light is going to pass from the light source and further on, there is a field diaphragm here. The field diaphragm is basically an opening which is going to control what exactly is the amount of light that can be allowed to pass further on. After a field diaphragm, we have a condenser aperture diaphragm, which also controls what is the amount of light that is going to pass from the condenser lens, which is kept here. A job of the condenser lens is basically to actually focus or condense the light which is coming from the light source onto your sample and illuminate your sample perfectly, uniformly. Now, I will make another video on how the uniform sample illumination is important for our microscopy, and you see in that case how do we align these condenser lenses to actually achieve uniform illumination. But for this point in time, I will stick to how and what all different type of components of the light microscope are. So after the condenser lens, there is a condenser aperture diaphragm, which is basically going to control the amount of light that is going to travel from the condenser to the object or the specimen. So just after the condenser diaphragm, there is a stage here, and this stage is basically which is going to hold your sample here. So the sample is placed on a sample stage, and your objective just above that, the objective lens is there, which is going to visualize it. Now, if you see it carefully, an objective lens is not a single lens. It's a turret like this, which has basically multiple lenses here, which I'm marking in red. Now, a collection of all these different lenses itself forms the objective lens. And we have objective lens associated with different magnifications such as 100x, such as 60x, 20x, and whatnot. Now, just after the objective and between the eyepiece here, you have out of the eyepiece, you have an eye with which you can actually visualize the image. But between the eyepiece and the objective, you have something which is referred to as optical tube. Now, in this optical tube, you have placed a tube lens here, which is basically very important to create this parallel beam of light after the objective. And after that, this parallel beam of light which is coming out of the objective has to be converged so that it basically forms a real and inverted image inside the back focal plane of your eyepiece, and then this eyepiece can actually form a upright and highly magnified image which can be visualized by your eye lens onto your retina. I hope with all this introduction and with all these topics thoroughly gone into it, you can acknowledge how an infinity space is created between the pipes and the objective by introducing the tube lens, which is of great application in today's or modern day microscopy. Also, we were able to see how an optical trail in the compound modern day microscope is followed and how it looks like. So, I thank you all for watching this video and staying towards the end. Keep watching and stay tuned.